Okay, let's dive right into this explainer. Today, we're going to decode one of the oldest, most misunderstood, and frankly, most terrifying medical mysteries known to humanity. We are talking about rabies. And when I say old, I mean ancient. The official documentation of this disease literally dates all the way back to the pre-Mosaic Eshmona Code of Mesopotamia in the 23rd century BC. For thousands of years, this pathogen has stalked humanity, leaving behind a trail of fear and scientific fascination. So in this explainer, we're going to break down the exact biological anatomy of this killer, we'll unravel a famous medical myth that offered a false hope, and finally, we're going to look at the incredibly simple yet highly effective true cure. So the crucial point to understand right up front is the sheer stakes of this virus, 99.9%. That is not a typo, folks. That is the fatality rate of rabies once clinical symptoms appear. In the entire realm of human infectious diseases, there is absolutely nothing else like it. If a person is exposed to the virus and doesn't receive preventative treatment before the onset of symptoms, things like fever, confusion, or hydrophobia, the disease is virtually inevitably fatal. It's a biological point of no return. It establishes this ruthless timeline where any delay can be deadly. And because of that terrifying fatality rate, the global burden remains just devastating. Despite being effectively eliminated in places like Western Europe, North America, and Japan, this is still a severely neglected tropical disease. It claims around 60,000 lives every single year, primarily in under-resourced areas across Asia and Africa. And what adds immense gravity to this statistic is that 40% of those victims are children under the age of 15. Because of their shorter stature and, their natural curiosity around animals, they're far more likely to sustain severe bites in high-risk areas like the head or neck, which dramatically accelerates the timeline of the infection. Now, let's get into the literal anatomy of this killer. This is the rabies virus, a member of the Rhabdoviridae family. It has this highly distinct asymmetrical shape. Basically, it's a 180 nanometer bullet-shaped particle. One end is rounded and the other is flat. The surface of this lipid envelope is densely covered in about 400 of these glycoprotein spikes, which act like microscopic lockpicks designed to infiltrate host cells. And inside that bullet casing sits a tightly wound negative-sense single-stranded RNA genome that codes for just 5 proteins. It's structurally simple yet beautifully engineered for absolute biological devastation. To cause such widespread destruction, the virus embarks on a terrifying, highly coordinated 4-step microscopic heist. It always starts with the initial exposure, usually an animal bite, that directly pushes the viral particles deep into the muscle tissue. From there, it immediately seeks out the neuromuscular junction, initiates a rapid climb up the spinal cord, and finally achieves its ultimate goal, a total hijacking of the central nervous system and the brain. The breach itself begins at the neuromuscular junction. That's the exact microscopic point where your nerve endings meet your muscle fibers. The virus utilizes those glycoprotein spikes we just talked about to bind to very specific receptors on your muscle cells, specifically targeting the nicotinic acetylcholine receptor. It concentrates rapidly at this junction, quietly slipping directly into the peripheral nervous system. It's essentially a stealth breach, perfectly calculated to avoid triggering the standard alarm bells of your body's immune system. Once it's safely inside the nerve, the virus faces a massive logistical problem. It needs to get to the brain, which could literally be feet away, but obviously a virus can't just swim there. So it utilizes this brilliant, albeit sinister mechanism called retrograde axonal transport. The rabies virus literally hijacks your own cellular motor proteins, specifically a transport protein called dynin, to travel backward along the microtubule networks inside your axons. It seamlessly turns your nervous system's internal communication highway into its own personal express route to the central nervous system. The tragic reality of this stealth invasion is how highly evasive it remains. As the virus spreads transsynaptically from neuron to neuron, traveling methodically up the spinal cord, it remains virtually invisible. Because it's hiding strictly inside the nerve cells, the host's adaptive immune system largely doesn't even know it's there. By the time it finally reaches the brain and begins massive, uncontrolled replication, triggering those classic clinical horrors of rabies like severe hallucinations, paralysis, and that intense physiological fear of water we call hydrophobia, it's too late. The virus has already established dominance. Because this late-stage infection is a known biological death sentence, the medical community has desperately searched for any possible treatments. For years, a medical myth circulated heavily about a miraculous induced coma treatment called the Milwaukee Protocol. It made international headlines when it supposedly saved a teenage patient named Gina Gies. The foundational idea was to put the patient in a deep, brain-protecting coma, while their immune system naturally fought off the virus. But we have to ask the hard scientific question here. Did the protocol actually cure her rabies, or was there another factor at play? Well, intensive investigation over the years reveals that Jenna Gies' survival was likely a massive immunological anomaly, rather than a reproducible triumph of the Milwaukee protocol itself. First off, the bat that bit her was never recovered, leaving open the very real possibility that she was infected with a much weaker, attenuated variant of the virus. Furthermore, upon admission, she already exhibited an unusually robust native immune response, with incredibly high levels of neutralizing antibodies already present in her spinal fluid. That is something almost never seen in fatal rabies cases. Her body was fundamentally already fighting off a potentially weaker virus before the coma was even induced. The tragic proof lies strictly in the data that followed. In the 2 decades since that initial index case, doctors around the entire world have tried desperately to replicate this protocol, and there have been at least 64 documented cases of the Milwaukee protocol utterly failing. In one major compilation specifically tracking 31 patients treated with this intensive care regimen, all 31 died. Across all data points, there have simply been no reproducible, verifiable survival outcomes linked to the treatment. And clinging to this futile treatment is actually actively harmful. Rigorous scientific appraisal shows the protocol is inherently flawed at a pathophysiological level. Think about the biology of it. Inducing a deep coma with heavy barbiturates inevitably causes profound immunosuppression. So during an active, aggressive viral infection of the brain, the protocol actually suppresses the host's natural immune response. This actively accelerates viral replication and spread. Plus, keeping patients locked in intensive care for weeks leads to severe cardiac issues, respiratory toxicities, and fatal secondary ICU infections. Modern medicine is now aggressively moving away from this false hope. So if intensive care and post-symptom treatments like the Milwaukee protocol are a definitive dead end, how do we systematically stop a disease with a 99.9% fatality rate? Well, we have to focus entirely on the true source of the problem. While we hear a lot about bats and raccoons in places like North America, the global reality is starkly different. 99% of all human rabies cases are directly transmitted by infected domestic dogs. To stop rabies at the human level, we absolutely have to address canine transmission head-on. The reality of this disease is actually incredibly empowering when you compare these two outcomes. On the one hand, we see that once symptoms appear, intensive care is completely futile and physiological suffering is high. But on the other hand, we have the definitive true cure, post-exposure prophylaxis, or PEP. If a patient receives immediate PEP before that bullet-shaped virus has a chance to hijack the nervous system through retrograde transport, the disease is essentially 100% preventable. It is an absolute, guaranteed lifesaver when administered correctly. This means our true cure isn't found in the complex, expensive machinery of an intensive care unit. It's found strictly in the power of targeted prevention. It means prompt wound washing, a timely series of specific rabies vaccines, and passive immunization through immunoglobulins if needed. But strategically, it means mass vaccinating dog populations at the source. Mass dog vaccination serves as the ultimate shield for humanity. In fact, analytical models estimate that if we reallocated just 10% of the financial resources currently spent worldwide on emergency post-bite treatments, we could fully fund national veterinary services to eliminate rabies in dogs entirely. Which brings us to the most crucial point, and I'll leave you with this final thought to reflect on. We have the complete biological knowledge. We've mapped the exact anatomy of this 180-nanometer bullet-shaped killer. We understand precisely how it hotwires your nerves to travel up the spinal cord, and we have the exact 100% effective tools to prevent it. If just a fraction of current emergency spending could eradicate this ancient terror at its source in dogs, why does this fully preventable disease still claim 60,000 lives a year? We have the tools, the data, and the cure. It's simply a matter of execution. Thank you for joining me on this explainer, and keep questioning the world around you.